Good afternoon. Welcome to UT Brainstorms. Yay, so thank you for coming. So this is our 20th UT Brainstorms, and we were adding it up last night. That means approximately 8,000 audience members now total, so thank you for being a part of that. For how many of you is this the first time? This is always a fun, wow, it just keeps, thank you for coming, okay. And you all heard through Facebook, didn't you? Yeah, so, no? Okay. You have to come and tell us how you found out if it wasn't because we're always wondering how to reach more people. But anyway, so those of you who have been here before have heard this, so apologies, but for the newbies, this is sponsored by the Department of Neuroscience. My name's Mike Mock. I'm the chair of the Department of Neuroscience, and this is just something that we started a couple of years ago with the feeling that as a world-class neuroscience department, it was just something we should do to make ourselves available to the community and to share what we know about neuroscience and to listen to what you have to say. So we have conversation in the title of all of our talks for a reason. We want to tell you and share with you what we know, and we learn a lot from what we hear from you. So we want to make sure it's a conversation, and so the way that's going to work then is our speaker's going to go, and then when he's done, we're going to assemble a panel up here, and then there's a number that you can text. We've joined the 21st century. And you'll text your questions, our panelists will pick them, they'll get zapped over here, and then they'll be on the screen, and we're just going to be state of the art. So anyway, I've looked forward to this particular Brainstorms for a while now, in part because the topic of Parkinson's disease is important to me. My mom was diagnosed quite some time ago. She's doing fine, thank you. And in part because just down the road at UTSA, we have one of the world's authorities on the, well, actually, honestly, he'll object to this, the world's authority on the basal ganglia, which is the part of the brain that's under attack in Parkinson's disease. So our speaker today is Dr. Charlie Wilson. Known Charlie for a long time. He's a great guy. He's a great speaker. He's in the Department of Biology at UTSA. He's the director of their neuroscience program, and he is holds an endowed chair there. And I can assure you that he's a great speaker. You're going to see that, but I can assure you he is widely respected as, and I certainly feel this way as well, as just an old school kind of scientist. When he publishes something, man, you know it's true. And this is a hard combination, incredibly innovative and creative scientist. And so it's a great pleasure to have him here. So please help me welcome Dr. Charlie Wilson.
Thank you very much, and thank you, Mike. That was really nice. And I'm going to talk about Parkinson's disease. And just for one slide, I'm just going to talk about what Parkinson's disease is. I think probably because Parkinson's disease is a very prevalent disease, it affects about 0.3% of the entire world's population. That's about 1% of people over 60. There are about 1 million people in the United States with Parkinson's disease. There are about 10 million people in the world with Parkinson's disease. So it's very common, and everybody knows somebody, either in their family or a close friend or both, who have the disease. It is hard to know what the cause of the disease is. My nephew got diagnosed a couple of years ago, and his first question for me was, why me? And for the most part, we don't know the answer to that. It isn't something, as far as we know, I told him, it isn't something you did. But there are a small percentage of cases that really are familial, and there are familial forms of Parkinson's disease. And these are very illuminating scientifically, because we might be able to identify a gene that causes Parkinson's disease, and it may not cause everybody's Parkinson's disease, but if we knew the pathway by which a particular gene causes the disease, that would help us to understand the disease itself and the disease process. And so that's an important aspect of it, even though it's maybe medically not super important because it's a really small number of people that have the genetic forms. So the symptoms of the disease are numerous and varied and different. Patients have different symptom sets, but the classic ones that are good for diagnosis of the disease are resting tremor, and this is usually what brings people into the clinic, is they have a tremor in a limb, usually just on one side, and it's worse when the limb is not in motion compared to when it is. So that's resting tremor. Rigidity is just stiffness, especially in the limbs, and they resist being moved. Bradykinesia just means moving slowly, so everything that one tries to do happens more slowly than you intend for it to and that you would normally do. And postural instability is probably a result of some of those things. So because of the rigidity and the bradykinesia, the posture is uncertain and balance is affected, and falls are a real danger for Parkinson's patients. And they have a kind of gait. This is an old image drawn in 1886 showing the Parkinsonian, this is an advanced case of Parkinson's disease, and the sort of Parkinsonian posture, which is a kind of posture defending against postural instability. It's one that increases stability, and Parkinson's patients often take very small steps, and part of that is an adaptation to their postural instability. In some patients, there's freezing, which is a mysterious kind of symptom, because walking along and come to a doorway and freeze and not be able to go through the doorway. And then getting unstuck, getting unfrozen becomes an issue, and patients have tricks sometimes that get them unfrozen. Maybe I saw a film of a patient who carried a little rubber ball, and when he would freeze at a doorway, he would drop the ball, and it would bounce on the other side of the doorway, and he could step through and get it, like that. Isn't that mysterious and weird? So it isn't so much that the mechanism of the body isn't working, because under some circumstances, it does. It's that just for some reason the control of that mechanism is lost, and patients sometimes describe themselves as imprisoned in their body. They know what they want to do, but for some reason they can't get their body to do it. Micrographia is very small handwriting, and so this is something that most Parkinson patients will recognize right away. When I try to write, I write really, really small. Some patients have dystonia, which is a sort of cramp maybe in a hand or the foot, where the muscles just contract because of rigidity, but they contract in an uncomfortable, sometimes really painful position. And then anosmia, which is just a loss of the sense of smell, and sometimes happens really early in Parkinson's disease and is a kind of precursor of the disease and happens a long time before the motor symptoms do. And the relationship between all of these symptoms is not completely understood, and they do not all necessarily arise from a single part of the brain, a single brain dysfunction. So part of the mystery is figuring out how all these symptoms fit together and how they could somehow be caused by the brain changes that we know happen in Parkinson's disease. Now I want to start with a little disclaimer. So I'm not a physician, I'm not a neurologist, I do not see patients. My knowledge of Parkinson's disease, my exposure to Parkinson's disease is like almost everybody else. I've had close friends and family members who have the disease. Do not treat people with disease, and I can't give any medical advice. I wouldn't dare try. But what I study are the brain mechanisms that give rise to the disease, and that's what I think I'm qualified to talk about, and that's what I'm going to restrict myself to talking about, those things. And in fact, there's been a sequence of treatments that have been discovered, and each one of them arises because of a sort of breakthrough in understanding. And so I'd like to go through those breakthroughs in understanding and the treatments that came from them, and maybe we'll end with some sort of notion about the future of Parkinson's treatments. So The fundamental finding that began the scientific study of Parkinson's disease was this. And these are sections through the brain of a control and a Parkinson's disease patient in the midbrain. And it's very hard for me to give you any kind of perspective on this, but this section is through something about maybe the size of a quarter in diameter. It's been blown up a lot. And if you look at this, you can see these black structure on the control side, and that is a black pigment that is in some cells in a part of the brain called the substantia nigra. That's called that because there's black pigment in the cells. And long time before the thank you. A long time before the cells were identified that have the black pigment, there we go. That black pigment was observed. And neurologists studying the brains of Parkinson's patients saw the black pigment is missing. And that was sort of the first discovery of exactly what's going wrong in the brain in Parkinson's disease. And in fact, this became the definitive diagnosis for Parkinson's disease, and really you couldn't have a definitive diagnosis while you were still alive, because to see that you would have to cut the brain. And so diagnosis came post-mortem, even though doctors would say, I think you have Parkinson's disease and we're going to try to treat you for it. Now there are, actually, you might be glad to know this isn't really required for a definitive diagnosis anymore. But the discovery that those cells were missing was really the key to the very beginning of treatment for Parkinson's disease. And I should say, there are lots of neurological diseases, degenerative diseases in the brain that cause cells to die and disappear in the brain. And most of them are devastating, destructive diseases, and almost none of them have any treatment whatsoever. And Parkinson's disease stands alone in that there are so many treatments and very effective treatments for Parkinson's disease. And you might wonder, why is that? Why are there so many treatments for Parkinson's disease and there are no treatments for Alzheimer's disease? And that's a very good question. So, and I don't know how to give you a simple answer except to walk through the science that led the step-by-step to each of the treatments for Parkinson's disease, and we could kind of see where they came from. So where do medical treatments come from in the first place? And sometimes we just think medical treatments come from some smart doctor who had a hunch. And he had a hunch, I should try this, and maybe it would help. And they try it, and it helps. And sometimes they try it, and it doesn't, and they discard that approach. And then some smart doctor with a hunch tries it, and it works. And I think it's true. I think that's sort of roughly how it happens. But where do the hunches come from? And the hunches don't come out of thin air. The hunches come from models of how the disease is working. And the models come from science. So the science doesn't directly create the treatments. The scientists create these models for what's going on, and then physicians create treatments from those models. And so that's what we're going to see are some of these models. So in the 1960s, Arvid Carlson in Sweden discovered that the cells that contain that black pigment are the cells that make dopamine for the brain. And dopamine is a neurotransmitter, and it's synthesized in those cells, and almost all the dopamine in the brain comes from those cells. And so that led to really the first model of Parkinson's disease in the 1960s. And the little flowers are just for historical context, and they don't really have any function in the model. And the idea is that the brain is a vessel that contains a certain amount of dopamine, and it should have this much, and in Parkinson's disease it doesn't. Now that may seem like a silly model, but it immediately tells you what you want to do. And of course it's not true. It's not a true model of the brain. That's not what the brain is. But we use this kind of model of the brain all the time. How many times have you heard somebody say, depression is an imbalance between chemicals in the brain. Right? Well, this is exactly the model they're using. There's some chemicals. They're sloshing around in there. They have to be in the right proportion to each other, or else something goes wrong. So even though it looks like a silly model, and it's not a true model of the brain, it is a model that we use. We use this. We think like this. And Einstein said, when talking about explanations, that they should be as simple as possible, but no simpler. And so this was, at that moment, it was the simplest possible model, but it told you what to do. So the right thing to do is to fill the brain with dopamine. Somehow we've got to get more dopamine in the brain. Now, how do you do that? And you could start by saying, well, why don't we just give our patients some dopamine? We could feed them dopamine in their food, or we could inject them with dopamine. But the fact is, dopamine won't enter the brain. It doesn't cross the blood-brain barrier. So if you filled your blood vessels full of dopamine, it still wouldn't help your brain. It wouldn't fill the brain. So how do you fill the brain? And in the 1960s and 1950s they weren't a great time for neuroanatomy, I'll tell you that, but they were a really good time for neurochemistry. And one of the things people were super at during that period was discovering the metabolic pathways that would give rise to the chemicals that mattered in the brain. And one of them was dopamine. So this is great discovery of the 1960s, scientific discovery of the 1960s. Is complicated, but this one led to the first ever treatment of dopamine. So here's dopamine in the biosynthetic pathway. This is the dopamine molecule. This is where it lies. And these names are the names of enzymes that facilitate chemical reactions. And so these enzymes are themselves proteins, and so tyrosine is a common amino acid. It's an ordinary amino acid. It's present in food, and it's present in the brain in pretty high quantities, and it's synthesized from another amino acid, phenylalanine. And both of those enter across the blood-brain barrier with ease, and the brain has plenty of both of those amino acids in them. So you might, so if you start with that one, then there is a enzyme, tyrosine hydroxylase, that will help turn that into this molecule, which is dopa, and the L isomer, levodopa, people sometimes call it. And then there's another enzyme that turns levodopa into dopamine. And actually, dopamine is turned into norepinephrine and epinephrine too by two more enzymes, which are also transmitters in the brain. These norepinephrine and epinephrine are also. So if we want to increase dopamine, we could say, why don't we give our patients tyrosine? Because dopamine isn't going to make it into the brain, but tyrosine will. So if we increase tyrosine, maybe we could get more dopamine in the brain. But it turns out that in every one of these pathways, one of the enzymes' job is kind of gatekeeper. And that enzyme's activity is controlled by feedback loops from other molecules to make sure that you don't make too much of anything, especially dopamine. You don't want the brain making too much dopamine. And the brain is going to control dopamine synthesis, and it does it by controlling this step, tyrosine hydroxylase. So if you feed yourself lots of tyrosine and it gets in the brain, you still won't have more dopamine. What about this one? If we get around this gatekeeper, this enzyme doesn't care. It'll just be cranking out dopamine as fast as it can if it has enough L-dopa. And so the idea was, okay, let's give patients L-dopa. That will raise the level of dopamine in the brain, and that should help. And of course, as you know, that did help, and that does help. And many people take L-dopa today, and it really does help them. There is a little problem with L-dopa, and that is that you're loading the rest of the body, not the brain, full of L-dopa, and it's all got this enzyme dopa-deep carboxylase 2, and it's going to start turning all your L-dopa into dopamine before it gets to the brain, and then dopamine won't get into the brain, and so that keeps it from working. So what you need to do is to find a way of inhibiting this enzyme, but not in the brain, but only in the rest of the body. It sounds tricky, but it actually is possible to do. So you have an enzyme inhibitor that doesn't cross into the brain, And so you can inhibit that enzyme everywhere except the brain, and then you can take L-DOPA, and that will raise dopamine in the brain. And so that stuff is called Carbidopa. And so if you give a combination of DOPA and Carbidopa, then that will raise dopamine levels in the brain without wasting it by cranking up dopamine levels out in the rest of the body. So that stuff is what is the most common pharmacological treatment for Parkinson's disease. It's usually called Sinemet or something like that. It's a combination of L-DOPA and Carbidopa. So that was a huge breakthrough, and it worked. And the fact that it worked reinforced the model. It reinforced our trust in this as the synthetic pathway for dopamine. It reinforced our trust in the idea that the brain needs dopamine to come up to its fill level in order to work properly. And so for the moment, at least in the 1960s, that meant we have a treatment for Parkinson's disease. There's never really been one before. Now, there have been lots of other pharmacological treatments for Parkinson's disease designed, and many Parkinson's patients take a host of different ones that do different things, and every one of them has a cool story of discovery like this behind it, where people worked out biosynthetic pathways, how dopamine acts on the neurons, and designed molecules that would intercede in that process and then tried it and found out what worked. And so that's the basis for everything that we do pharmacologically. But L-DOPA is not perfect, and there are a couple of things that go wrong with the DOPA treatment. For one thing is the longer you go taking L-DOPA, the less effective it is. And so doctors often try to delay L-DOPA treatment for Parkinson's patients for a long time, because once you start taking L-DOPA, the brain is going to change in response to that, and the change is going to sort of run out the clock on L-DOPA's effectiveness. L-DOPA doesn't just suddenly quit working all at once, but it has a tendency to just become sort of an on and off phenomenon. So I'm having a lot of symptoms, I take DOPA, now I feel better for a while, but then the amount of time that I stay feeling better, which doctors call the on period for L-DOPA, that gets shorter and shorter. And so the Sinemet dose gets raised in order to overcome that, and then at some point some side effects of L-DOPA are seen. And those limit then how much you can raise the dose further. And so physicians who use L-DOPA on a regular basis became gradually dissatisfied with L-DOPA as an ultimate treatment for Parkinson's disease. And so we would want to know more about it. Why is it that L-DOPA treatment becomes less effective over time? And of course, the answer to that is that all of these molecules exist inside the dopamine neurons, and the dopamine neurons are degenerating. So if there were no dopamine neurons around, then there would be no neurons that have these enzymes to make dopamine, nobody to use that dopamine. And so as the disease progresses, DOPA becomes less effective because the dopamine neurons are required to make that drug work. And there are fewer and fewer dopamine neurons available. And so to go to the next level in trying to understand the treatments, or what the next generation of treatments ought to be, we can't just view the brain as a vessel with a certain amount of dopamine sloshing around in it. We have to know where in the brain is dopamine, how is it acting, why is it that the loss of dopamine causes the symptoms, and so on. We want to know where and how dopamine works in the brain. So the 1970s and 1980s were great times for neuroanatomy, and neuroanatomists learned a lot about the connectivity and structure of the brain. And so I want to give you like a really quick neuroanatomy lesson. People sometimes complain that neuroanatomy is boring and tedious, and I can't understand that. I think that's so wrong. Neuroanatomy is so cool and intricate. Imagine this very complicated, intricately, carefully designed machine with many, many parts in it, and now you want to figure out the machine, but you want to figure it out without ever figuring out what the parts are. Like, you're going to fix a bicycle, you don't know the difference between the chain and the handlebars. So let me just bear with me for a second. So this is a little diagram of a human brain, and this outer part is the cerebral cortex, and it's been made almost transparent, and it's not even really shown fully to scale because we don't care about the cerebral cortex which at all when we're thinking about Parkinson's disease. And underneath it, this blue stuff is the ventricles. This is a fluid-filled space in the brain. And the interesting parts for us are this green part, which is called putamen, and this, what is that, salmon-colored sort of part, which is the caudate nucleus. And together they make up the striatum, so we can think of them collectively as the striatum. And then there are these tiny little places underneath. So purple one, which you have to wait for it to come back around, but the purple one is globus pallidus, and it really has two parts, but we just have to mentally divide this purple part in half. And then this pink place there, which is the subthalamic nucleus, and then this gray one, very small part there, is the substantia nigra, where the dopamine cells themselves reside. And so these are the parts of the brain that matter for us. And in the old days of neuroanatomy, people were content to know these structures. But the thing that made it the 1980s special was the discovery of how they're connected to each other, because these structures don't act alone. They act together in groups. And so for the basal ganglia, we can sort of see some of the connectivity of the nuclei of interest. So this is half of the brain. So I'm showing you this half of my brain. Let's just look at this right half. And then I'm cutting a section like this. And you're looking at that face on at this half of my brain. And the outer part here is the cerebral cortex. And this is the putamen, which was that green thing. And this is the caudate nucleus. It was a salmon thing with this sort of tail, but we've cut through it. This is the globus pallidus, then the subthalamic nucleus, and the substantia nigra. So these are the players in the basal ganglia. And the key thing that we needed to understand to go to the next level of thinking about Parkinson's disease is how are these nuclei related to each other functionally. And each one of them, each part of the brain consists of individual cells, many, many cells, each one of which has a receptive surface, which is its dendrites on which it receives signals from other neurons, and then an output structure, which is its axon, which we can think of as sort of like a wire that carries its signal out and delivers it to other parts of the brain. So, for example, in the cortex, there are many cells who send their axons into the striatum, where they release transmitter substances and pass signals from the cortex to the striatum. And then the striatum sends signals to the globus pallidus and to substantia nigra and to subthalamic nucleus. And you can see it's very complicated, and in fact, this is only, I've left out some of the connections because otherwise it becomes kind of a mess of wires, like a real thing, like the kind of electric circuit we would build, that if you just open it up and look at it, you just see it looks like wires are going everywhere. Well, the brain is like that too. And but these are the major pathways, the biggest pathways, and the ones that we know have something to do with Parkinson's disease. So the cortex makes connections in the striatum to two parts of the globus pallidus, and these two parts are important. So if you're fading on me, wake up for a second just to get these two parts. The external part of the globus pallidus, and then the internal part of the globus pallidus. And they're going to turn out to be different players, even though they are so close together, and they both have the same first two initials. And the subthalamic nucleus. Now, most of these structures in the striatum are inhibitory. That means that the signal that they send to the next neuron tells it to be quieter rather than to be louder. It causes it to have less activity rather than more. And most of these structures are full of neurons that are constantly active on their own. They're just constantly releasing transmitter and making electrical signals, and then they receive inhibitory signals that cause them to slow down or stop for a moment. And I'm going to show you some of that happening. So this neuroanatomical knowledge was enough in 1989 or so to make the next version of the model of the basal ganglia. And this was made by Roger Albin and John Pinney and Ann Young, all of whom are sort of giant names in the neurology of Parkinson's disease field. And then the model was elaborated and redrawn and represented and many, many times by many people, and the one that I'm showing you is a relatively recent, for this model, incarnation of it, but it's basically got the same pieces that Pinney and Albin and Young presented in 1989. So the idea is that the cortex is making the striatum, and we'll just use the putamen and not worry about the caudate for this, the striatum, making the striatal cells fire more, making them more active. And that's why this arrow is drawn white. White in this means I make you more active. And these arrows are all black because they are inhibitory, and so the striatum inhibits the globus pallidus, and it inhibits GPE, that's the external segment, and GPI, which is the internal segment. And the subthalamic nucleus is an excitatory one, so it has a white arrow. And you can see that what's happening is if we follow this from the cortex, we say cortex makes putamen fire faster, putamen makes GPE fire slower. Cortex made this fire faster, that makes this fire slower, and so that means this is going to fire less, and that's going to make this one fire more, and that's going to make this fire more. This one, the cortex makes this one fire more, and then it makes this one fire less. So if you follow the cortex through these two pathways, what you see is that this one is going to make, it's got two negatives and a positive, so that means it's going to be a net positive effect, and this one's going to be a net negative effect. So they're doing opposite things to each other. So this one, which is usually called the direct pathway because it goes straight, it is doing the opposite thing from this one, which is called the indirect pathway. You might think, why would the brain make two opposite signals and send them to the same place? And of course, the idea is it's not going to do that. Sometimes it's going to do one way, and sometimes it's going to do the other. So it's like a balance between this direct and indirect pathway. And at any one moment, one of them is winning and the other one is losing. So we think of this as kind of a seesaw, where the direct pathway is on one side and the indirect pathway is on the other, and the output of the basal ganglia is the result of that seesaw balance. So what matters, so let me just tell you what the seesaw does. So if these cells, the GPI cells, fire more, then that's going to inhibit this structure, the thalamus, and so there'll be less activity in the cortex. So this route is going to inhibit the inhibitory cells, is going to release the cortex, and this route is going to stop the cortex from doing whatever it was doing. So people think of this as a go pathway. The cortex said, I'd like to make this movement, and if the direct pathway wins, then that means, okay, go ahead and make the movement. And so it's considered kind of a go pathway. And the indirect pathway is going to inhibit the release of that movement, and so it's kind of a stop pathway. Now, let me just tell you, the brain doesn't spend that much of its structure and effort in neurons just making a stop-go signal, which is like it's almost like imagining the brain as a vessel that holds dopamine. But just like that model, this model has predictive power, and it suggests that we might try stuff. Even if it isn't exactly right, maybe it predicts something that's right. And so one of the things that it says is, if dopamine's job is to adjust the seesaw. And so dopamine decides whether we go or whether we stop. Then dopamine inputs to the striatum could be telling it, go, stop, go, stop, go, stop, go, stop. And when you don't get enough dopamine, it says stop all the time. So a real good friend of mine who is a neurologist told me that Parkinson's disease is a permanent state of motor disappointment. So the motor cortex is saying to the basal ganglia, how about this? Let's do this. And the basal ganglia says no. And so the cortex says, well, how about this? The basal ganglia says no. And it's just basal ganglia says no to everything. So that's the idea behind this model. And you could even imagine that some of the peculiarities of Parkinson's disease, like if a Parkinson's patient is having trouble walking, you can sometimes give them a laser pointer, and if they point at a place on the floor, they can step out to that place onto the floor. And I don't know if you've ever seen this, but this is something that neurologists often offer patients. So you wonder, well, why would that work? And maybe that is offering some new, unique option that the basal ganglia doesn't get around to saying no to. Okay, so their idea was the dopamine cells die, and now this indirect pathway is overactive. That's why it's broad. Broad arrow means more message traffic going in that pathway. And so that means all of these do less. That means more inhibition, and the cortex is being told no all the time. So this was their model for Parkinson's disease, introduced in 1989 and continues to this day. People continue to work on this model and think about how to improve it and what it suggests we should do. So, of course, I told you models are good because they give doctors hunches about what to try. And so this particular model was produced by neurologists in the first place. And by the way, a lot of this was very fanciful. The anatomical connections were there, but the idea that dopamine causes one of these to go up and one to go down and all of this was mostly just made up. There are lots of things that we knew about the structure of the basal ganglia, but we didn't know very much about the function of the basal ganglia. So if you're going to make a model at that time, you basically had to make up all the functional parts. And so that's why it's a model. I mean, if it was all right, it wouldn't be a model anymore, it'd be the actual brain, right? So, but it gave an idea, and the idea is that maybe we could make a lesion in the GPI, actually destroy a place, like fighting fire with fire. There's a part of the brain that's dying, and that's what's causing this disease. And the disease isn't really preventing people from moving, it's just causing the brain to choose not to move. And so you could disable the part of the brain that's making that wrong choice, and movements would come again. It's a kind of a risky thing. Maybe you would think it was a dangerous thing to try, but they thought maybe we should try making a lesion in the GPI. And if we did that, then we would reduce this. So this great big wide arrow, which is inhibition, the no signal, the stop signal, we'd reduce it and make it smaller. And now that would release the cortex to do more of the stuff that it wanted to do. Maybe that would work. And if that's right, you sure don't want to make a lesion in the GPE. Because if you make a lesion in the GPE, then that's going to relieve the subthalamic nucleus, this STN, and it's going to excite this and make things worse. So a lesion in the GPE would make things worse. A lesion in the GPI would make things better. And that was just a hunch based on that model. And I told you a lot of the stuff in the model was just made up. And so you might think, I don't want to jump to that kind of conclusion. But in fact, it worked. So to do this kind of thing, you have to do surgery on a patient who's awake. So because what they had to do was find just the right place in the GPI. And so what that meant was that you could image, you can try to image the brain well enough to put an electrode in there to make a precisely placed lesion. But imaging at that time wasn't, when this started in the early 1990s, was really not good enough for that. In fact, you might say I think imaging is still not really quite good enough for that, brain imaging. And so once you think you're in about the right place, you needed to be able to record the electrical activity of the neurons and study them and look for what's different about them to tell you where you are. And to do that, you needed to have patients trying to make movements and making movements so that you could get the neurons to fire in relation to movement so you could see where am I in the right spot. And they also would stimulate places in the brain and see what kinds of effects that had on patients' movements. And so you needed a patient who would stay awake through a long, grueling surgery in which electrodes are being placed in the brain and measurements are being taken, but no decision has yet been made about exactly where we are and what lesion we're going to make. And then at some point, the careful study of the basal ganglia activity in this particular person, it gives a very precise location to where we should make the lesion. And then they start making the lesion and they watch what it does to the symptoms. And as this lesion gets made, the patient's symptoms should stop. And they do. And so when that first was seen, you can imagine the neurologists and neurosurgeons doing that really felt the excitement of discovery associated with the sort of scientific excitement of discovery, but also the discovery of a new therapy that would really help people. And you can see it's a very elaborate operation. The patient is an active part of it. And this complicated structure back here is a frame that allows them to place electrodes in known places in the brain relative to the imaging that they've been able to do. And it comes off after the surgery, so you won't have to wear that. So here's some examples of lesions that were made that actually worked, and now we have to look at these images the way the neurosurgeons and the neurologists have to look at them. That this is an image through the basal ganglia, but it's been cut in this plane, which is sort of looking straight down on the top of the head kind of plane. And this is imaging, so the patient brain is being imaged while they're perfectly fine. It doesn't do any harm to them. This is a MRI image, like the kind you might get of a knee when you have a knee injury. And you can see the parts of the brain. So this is the putamen. This part is the caudate. This is the external globus pallidus. This is the internal globus pallidus. And so you can see the electrode was placed and it made a lesion in the internal globus pallidus. Very important not to hit here, but to hit there. And the rostral part, rostral means toward the nose, so that's the direction of the nose. So the very front part and near the center is the target, and so this is a good hit. This one, also a good hit, but with a little bit of damage to the GPE, you can see, which is not really what you are hoping for. And this was always done just on one side at a time. It was quite rare to do it both sides of the brain. But this would be done on the side that was the opposite side of the brain to the side of the body that had the worst symptoms. And so often one side of the symptoms, motor symptoms, are much worse on one side than the other. And so if you could bring the bad side under control, the patient would be very improved by that and would feel that their lives had improved a lot. And so it's surprising, maybe, I don't know how surprised you are, it's shocking to me that this works. And it does work, and it was tried many times, and it worked pretty well many times, although there were a couple of ways that it could go wrong. If the lesion went into GPE, the patient got worse. And that's just what you would have said from that model, even though the model is just a model. But you would have predicted that. And so when that happened, it was tragic, but it also proved that the model was making the right predictions about the treatments. So I have to tell you about something that happened about the same time. So about the same time these lesions were getting started, there was a neurologist in Northern California who encountered patients, young patients, who were drug addicts, who seemed to have suddenly acquired a very advanced case of Parkinson's disease. So just young people that walked in that said, last week I was fine, and look at me now, and they had cases of Parkinson's disease that you would normally not expect to see for many years after diagnosis. So these were very advanced cases. And it turned out they were all drug addicts, and when he found as many of these patients as he could, he discovered that they all had taken the same not illegal drug, but designer drug. Because in those days, there was a kind of cat and mouse game going on between these people who were making new molecules that biochemistry would tell you would act like drugs of abuse, but weren't. They were new molecules, and so they weren't against the law. And so you could make them and sell them, and people would take them, and they were exactly like taking morphine or heroin or something, as far as the drug addict was concerned, but it was legal. And so there was a lot of money to be made doing that. And then the state legislatures would make those drugs illegal, and then these chemists would come up with new ones. And so they were just in a cat and mouse game like that. Isn't that strange? But that's the way we are. So eventually, the state legislatures learned enough chemistry or so, to make a law that would cover all of them somehow. I don't know what the law says. Anything that makes you feel good is illegal or something like that. But that tragic event, the discovery of that molecule and the identification of that molecule, which is a neurotoxin for dopamine cells and which killed the dopamine cells in those drug addicts right away, overnight, all of them, in a big way. So that drug became an experimental tool of great value because that meant that you could give an animal a case of Parkinson's disease. Now, it's not perfect Parkinson's disease. It's not caused by the same thing that causes our Parkinson's disease, which we still don't know what it is. But we know it isn't this drug. So we know that it isn't the same in that way. And our kind of Parkinson's disease develops slowly over many years, whereas this one happens right away. So in that case, it's not exactly the same thing either. But it creates the motor symptoms that we're used to being the most serious symptoms of Parkinson's disease, and we could create those in an experimental animal, and then we could study the generation of those signals. And so this was an early study in which monkeys treated with this drug, MPTP, and then recording neurons in those structures to see if the firing rates of neurons changed the way the model said they would. So if the model says the arrow gets broader, that means cells should be firing more. If the arrow gets narrower, it means cells should be firing less. So you should be able to check. And remember, the rule was that in GPE, the cells should be firing more in Parkinson's disease, and in GPI, they should be firing less. Remember that? So here we go. So in GPE, the normal monkey, actually this is the exact same set of monkeys before and after MPTP, which makes for an experiment that's way better than you could ever do with humans because you can study the same monkey's cells. The neurons are firing at 60 spikes a second before, and after MPTP, they go up to almost 80. So that's exactly what you predicted. It's an increase. It isn't anywhere near as big as the increase you expected it to be. Remember, the arrows increased by 3 or 4 times, and this is a 20% change. So you might be a little disappointed, but it's in the right direction, and it was statistically significant, you know, and the kind of stuff that scientists really care about, that kind of stuff. So it was a thing you could rely on. It was believable. And then they gave the monkeys L-Dopa, and after a series of L-Dopa, days of L-Dopa therapy, they could then study the monkey in the L-Dopa off, that is, it's been a long time since L-Dopa state, and then they would give L-Dopa and look at the on state of the monkey. And you could see that in the off state, the cells fired pretty fast, and in the on state, they slowed down. In fact, they slowed down way more than they were in the normal. So isn't that great? That's reassuring. I mean, to the people who believed in that model, they thought, this is it, this is great. And GPI also. So the cells are firing faster, they slow down in MPTP. Remember, they should have gone way down to here, but anyway, they went down. And then in the off, they're down, and the on, they're up. And so that's very reinforcing to the scientists. But the people who did this experiment were actually a little bit disappointed because they expected much bigger differences. And also the firing rate changes, they weren't the biggest thing. They weren't the thing that caught your eye. The thing that caught their eye was this. So the cells, now I have to tell you about neurons firing action potentials. I've been just talking about activity of neurons and say they fire more and less, but now I have to show you what that looks like. So here is an actual recording, and it includes a neuron and these slow changes. And you can, these slow up and down changes, and the neurons are these fast spiky things. And now you can filter that data so that you get just the fast spiky things, or you just get the slow stuff, that's this red thing, which is this slow waveform. And this one they called the local field potential because it's the compound effect of many synapses happening at once. And this is called a single unit because it's one neuron firing. And so what they saw was the neurons normally would be firing continuously, and now they're firing in these bursts. And the bursts are kind of repeating. They're not perfectly repeating, but you can see they fire in a kind of repetitive way. And the repetitive way is predictable from the field potential. And you can look at what frequencies are in the field potential. And what they saw was in the off state, that's when the monkey was the most Parkinsonian, had the most symptoms. There's suddenly this big oscillation between 10 and 15 hertz. So the thing that was unexpected but was the most prominent thing was this huge change in oscillations in the nuclei. And so that started a whole new way of thinking about Parkinson's disease. Maybe it's not the change in rate, which is kind of moderate. Maybe it's the change in the pattern of firing, which is a huge change. And not only were individual cells firing in this bursty way, but if you looked at multiple cells at once, they were firing together. They were bursting at the same time. And that never happened in the normal monkeys. Cells would be firing independently of each other everywhere in the GP and in the subthalamic nucleus. And suddenly when you look in the monkey, the MPTP monkey, you'd see this synchronous activity. So here are a bunch of different neurons, and you can see these neurons are all doing this at about the same time. And this is the monkey in the on state after giving him L-Dopa, and you can see the neuron activity has gone back to less bursty and more uncorrelated across cells. So they plotted the correlations, and you can see normally there are very few correlations. In MPTP, now the correlations go up 3 or 4 times the number of correlations, much bigger change than the firing rate change. And likewise, in the off and on state, you get this enormous change in this correlated bursting activity. So what they were expecting was firing rate, and they got it, but what they got that they didn't expect was this change in firing pattern. Now, we don't really know what the firing patterns mean, so it's a little bit like the intercepting encoded messages. So I guess the NSA is supposed to be doing this. They intercept coded messages that are being sent by people all over the world as they're plotting terrorist attacks and that sort of thing. But they don't know the code. They can't, at least, according to the story I read, they can't crack those codes, so they don't know what the messages mean. but they can tell a lot just by studying how many messages one group is sending to another group. And just before something happens, this message traffic goes up. And so they can issue a red alert because message traffic has gone up a lot. They know something's about to happen. And they know about where it's going to happen because they know where the message traffic has gone up and between whom. But they don't really know the message. And we're in exactly that kind of situation. The neurons are sending messages to each other. We don't know the message. We can't read the message. But when they send more or less, we say there's more activity or less activity. And what we discovered by looking at firing rates was, well, message traffic tells you something, but the pattern changed. It isn't just that the amount of message is changed, it's that the pattern, the actual message, whatever it is that's being said, is a different message than it was before. So now what we really need to do is to understand those messages and figure out what is the meaning of the message that the neurons are sending to each other. So, of course, all that was done in MPTP monkeys. We want to study humans and understand things in humans because they may not be the same thing. So, but those experiments told some of the neurosurgeons and neurologists what they could do. So as they're putting electrodes into these structures in patients in order to make lesions, they should measure some electrical activity in these field potentials, the ones that have this oscillation in them. And they should look for that oscillation. Is that happening in people, or is that just an MPTP monkey phenomenon? And so here's an image during real surgery of some electrodes placed in the globus pallidus and in subthalamic nucleus. Subthalamic nucleus is down there. You can't really see it. We can't see subthalamic nucleus in images very well at all, in brain images at all, but they know about where it is. And so they went there and then they used electrical signals to tell where they were. And then they could look in the on and off state. So here's off, and you see there's that crazy oscillation that was seen in the monkey, and you see it in both of these two nuclei. And then in the on state, that is with L-DOPA therapy, the subthalamic nucleus switches from this really slow thing to this fast, noisy looking thing. That is what you'd see normally in a normal monkey would look like this, not like that. So the human Parkinson's patient has these oscillations just like the MPTP monkey does, or very close to it. So maybe that's part of what's going wrong. Maybe that it is not the total amount of message traffic, but this oscillation that is affecting movement. And so here's another experiment. This is from the same guy as the one I just showed you, Peter Brown, who's a real leader in this field, and he is recording in humans, and this is recording in the subthalamic nucleus of a human, and you can see this bursting. Doesn't that look exactly like what we saw in the monkey, MPTP monkey? And this is the field potential with this low frequency oscillation in it. And then, of course, these are patients who are awake and able to move their arms and talk to the surgeons. And so the surgeon is asking the patient to make movements, and the patient is having trouble making the movement. And at some point, the patient becomes sort of unfrozen and executes the movement. And this red stripe is that low frequency. It's that thing. This is another way of seeing that frequency as this sort of red stripe in this frequency range. And just before the movement begins right at that arrow, you see that low frequency oscillation is gone. It just stops. And then the movement is executed, and then it comes back. So that oscillation is the thing that's, okay, maybe not. The oscillation is very highly correlated with the symptoms, and in what looks like it could actually be causing the motor symptom, the oscillation itself. So if there was a way to disturb the oscillation without changing anything else, maybe that would be a great treatment. And so while they were thinking about that, a third lesion got considered, and that was a lesion in the subthalamic nucleus. And if you think about the subthalamic nucleus, it's a great place to make a lesion because it drives this region, the GPI, which is the one that's inhibiting movement. So if you could cut off the input from the subthalamic nucleus, make it quiet, the GPI would go quiet, and things would go back to normal in the cortex. So many people thought, we're making lesions in the wrong place. We should get away from GPI because it's too close to GPE anyway, and there's a danger of hitting GPE. We should make our lesions in subthalamic nucleus. And that was not doable. And the reason it was not doable is because humans sometimes get bleeding in the subthalamic nucleus or strokes just spontaneously. It doesn't happen very often. It's kind of a rare thing. But when it happens, there's a kind of motor syndrome that comes with that called ballismus, which is a sort of failure to be able to control ballistic movements. And it's quite severe. And so no insurance company wanted to pay for that kind of thing. And most, because it could turn out very badly, and most neurosurgeons shied away from doing it. But it was tried in the MPTP monkey. You don't need insurance to do that. And it worked. It worked incredibly well in the MPTP monkeys. And so there were some private patients with the means to pay for it themselves who had this done. Quite a lot of them outside the country and even a few inside the United States. And they had very positive outcome. And they did have ballismus, but it went away over a short period of time. And then they were really better. But it was impossible to sell to the authorities. And so the people who were really in favor of subthalamic nucleus lesions thought, isn't there something else that we could do to the subthalamic nucleus? And the answer was, actually, we can stimulate the subthalamic nucleus because the rules regarding what you can stimulate are different from the rules regarding what you can lesion. And so it was possible to stimulate almost anywhere without running into a lot of the regulatory stuff. And so they thought, well, let's try stimulating in the subthalamic nucleus. Now, stimulating sounds bad. We're going to make it fire more rather than less, right? But we know that if we stimulate some neurons really, really, really fast, that they can't keep up with it. They get kind of exhausted. They go into a state of block. And so if you stimulate them really fast, maybe we can just turn them off. And it will be like making a lesion. And better, because you could turn the stimulator off, they'd come back, and you wouldn't be stuck with the outcome, right? You make a lesion, you're stuck with the outcome. But if a stimulator, you could turn it on, turn it off. So if it doesn't work, we just turn it off. And so they tried that. And of course, the outcome of that was deep brain stimulation. And deep brain stimulation was amazing because it worked. And so the idea behind deep brain stimulation was really originally that. It was to turn off parts of the brain by activating them too much excitement, and they would shut down. And they tried it in the globus pallidus internal segment, where turning it off would be like a lesion, only you could reverse it, and in the subthalamic nucleus, and it was effective in both of those places. And so it was very exciting outcome, and many patients now have deep brain stimulation, and it is generally preferred over lesions, and most of the clinics that used to do lesions don't do them anymore because deep brain stimulation is better. What's better about it? One of the things that's better about it is the fact that you can turn it off. So if you give a lesion to somebody and then you wait 3 years and they say, well, I don't know whether it's really helping me in the long run or not, because I don't know what I would have been like 3 years later if I hadn't had the lesion. But with deep brain stimulation, if you're curious about what I would be like if I hadn't had deep brain stimulation, you can just turn it off and you can find out. And if it turns out that 5 years later, 6 years later, 10 years later, turning it off is bad, turning it on is better, then you think, well, deep brain stimulation is still working for me, at least some. And there are plenty of patients now that have had it for decades. So, you can go on YouTube and see these videos. Some patients post videos of themselves turning off their stimulator and then showing you their symptoms and then turning it on and showing you what they can do. One of them I watched a couple nights ago was somebody 8 years out, and he was still getting an enormous amount of relief from the deep brain stimulation. So there's something weird, though, about the deep brain stimulation, too, and that is when one then looks this is done in monkeys, but now it's even been done in humans, and you record, it turns out that it's not shutting the neurons down. It actually is stimulating them. And so you thought it was going to work because it shut the neurons down. That was your model. But it turns out it works, but that's not why. Also, if you can stimulate external pallidum, which should have made you worse, it works too. External pallidum works too. So that model that led you to try deep brain stimulation doesn't predict the results of deep brain stimulation. It was great. It got you to try it. But it's broken. Deep brain stimulation now tells you need a new model. You need a model that tells you what's causing the oscillations and the synchrony of activity, where that's coming from in the brain, why loss of dopamine causes that, and how deep brain stimulation can suppress it. So we know deep brain stimulation suppresses the oscillation. We know it suppresses the synchrony. We don't know exactly how that does that, but we know that it does activate. So here's the experiment done in monkeys. And I have to explain this. It takes a second, but I'm almost done with this talk. So if you can look through one more set of crazy figures from scientists. Here's a recording from a GPI neuron in a monkey, and the stimulation is in subthalamic nucleus. And this is looking at the neurons. And when we turn on the stimulator, so this is stimulation and then stimulating again. And you can see that right after the stimulation, there's this big period where the cell fires, and then it fires again just before, and then on the next stimulation, it's going to look just the same. So the cell has become entrained to the stimulation, and it's tending to fire at two different points in the period between stimuli. And if you look at turning the stimulator on and turning it off, you can see this is the firing rate of GPI neuron before stimulation. When it's on, and this is GPI neurons, when they fire more, the symptoms should get worse. But in fact, the symptoms get better. They're firing more while the stimulator is on. And so this is the puzzle. This is where we stand. We need a new model that can predict this, can explain it. That model that told us to try this was great. It was just as simple as it could be and no simpler. But now we've seen something that it doesn't explain. And it's an opportunity for us to go to the next level of understanding of what's happening in Parkinson's disease and make a model that predicts that, which will then inspire new treatments. And maybe this is something like it. It's based on oscillators, which have to have feedback. The old model had no feedback. So nothing ever went in a loop. Activity didn't go in loops, and loops are what make feedback. And so we've just rearranged the anatomy a little bit, and we can see that there are actually mini loops in the basal ganglia that could be making feedback. There's this loop between the subthalamic nucleus and GPE. The GPE neurons onto themselves. There's back and forth between GPE and striatum. Remember, the old model didn't even show this arrow that went from GPE to striatum. But we knew there were neurons that did that. They just got left out of the model because there was no function for them in that old model. But now that we're looking for a new model, we look at pathways that were left out of the old one in order to simplify it. And we see that they are enriching it with features that are going to build the basis for our next generation model. So thank you very much, and that's it.
Fantastic. Thank you, Charlie. Awesome. So we're going to assemble our panel, and I'll introduce them as we get set up. And we're going to get set up with our little electronic trick, I think. So you're about to see a text number that you can text your questions to. The questions will go over to our panelists, and they'll zap them to the screen, and then we'll answer, and then we'll just do that until it's time to leave. Okay? This is Diane Whitmer. She's an instructor in the Department of Neuroscience, and she has a background in basal ganglia and deep brain stimulation. And so please welcome Diane as one of our panelists. You know Charlie? I'll read the questions and then you guys go. How
Okay,
about
you read
that?
it.
So here we are, first question. Thank you. Thank you, Elena. So before I start, Elena Silva, thank you. She runs all of this for us. Thank you. And they're around. You see people with UT Brainstorms t-shirts. Most of them are from they're students from St. Edward's. We have this little army of helpers, so thank you to them as well. So there's your phone number at the top. And first question, have there been any studies correlating frequency of...
Fair question. I'll tell you, though, I don't know the answer to that question.
Okay.
I know that there are studies of the effect of Parkinson's disease on sexual activity, but I've never heard of one that had anything to do with predicting onset. And
Okay, good. Yeah, good. Good answer. It's a good answer. Okay. All right. Questionable question selections there, but okay. When lesions of GPI or subthalamic nucleus succeed in removing Parkinson's symptoms, is the improvement permanent as opposed to the L-DOPA treatment?
so this reminds me of something I really had intended to say and didn't. And it is that with lesions, patients still had to take their L-DOPA. And in fact, they didn't usually reduce their L-DOPA dose for very long, sometimes for a while.
Yeah.
But with DBS, patients almost always reduce their L-DOPA. I mean, maybe some of you have real experience and could answer this better than I do, but the way I understand it from what I've read is that most patients reduce their L-DOPA dose with DBS, and that it even happens that some patients quit taking L-DOPA. And so that was a very good sign about it. Now, is it permanent? That would require, permanent is a long time. So what I'll say is, for as long people have only, I guess the people that started getting DBS got it maybe 20 years ago, and some of them are still showing a lot of benefit from it. So 20 years from now, we can answer the question about 40 years. But the, and maybe you were thinking, when you asked this question, I would give you more answer than you asked for, but maybe you were thinking, you know, L-DOPA seems to create its own side effects. There's sort of a process in which L-DOPA goes from being really effective to being less effective. And is there something like that in DBS, in which the side effects get worse and the DBS gets less effective? And the answer is sort of no. People do have to get their DBS kind of reprogrammed and retuned from time to time, but it doesn't take more and more and more current all the time. And there are sometimes some side effects from DBS, but they usually show up right away. They're not something that develops slowly over time.
Okay, thank you. Is DBS effective for advanced Parkinson's patients?
You know, most, in the beginning, the only patients that got DBS were advanced Parkinson's patients. And so in the beginning, all of the successes were pretty much with patients that had run out of options, because the DBS surgery seems kind of radical. I mean, I don't know how you feel about it, but it strikes me as a radical thing to do. It certainly seemed that way to the neurosurgeons and neurologists who did it in the beginning. And so they didn't want to do it with patients that were responding perfectly fine to more traditional approaches. Now, I think people are getting DBS earlier and earlier in the disease. But yes, even very advanced patients can benefit from DBS.
How do you keep a patient awake during surgery?
I can address this.
You want to answer that?
Yeah, so you may know from neuroscience that there are no pain receptors in the brain. And so there's a little bit of flexibility in terms of the anesthesia that's administered during deep brain stimulation surgery. So, you know, the way to keep a patient awake is to not put them to sleep through anesthesia. And I'd say also that, you know, patients in this scenario are getting interaction from their neurologist. So the neurologist will be asking them to make these kinds of movements as the electrode is being lowered. And that's for the purposes of ensuring that the deep brain stimulator is in the correct region of the brain. It's a functional confirmation.
No? All right. Do Parkinson's symptoms vary, or do all patients conclude with the same level of severity?
I can take this. So Parkinson's is a very heterogeneous disease with, you know, some patients have freezing of gait while others don't. Some have a lot of rigidity and bradykinesia without tremor. Others are more tremor dominant. So both the types of symptoms and the severity varies greatly between people. Do you want to add anything?
I don't know, I have to remind you again, I'm not a physician. I don't see Parkinson's patients, but I watch YouTube videos about them. And I watched a YouTube video that Michael J. Fox did recently. He does a lot of really great YouTube videos on Parkinson's disease. And he went around and visited a bunch of different people who had Parkinson's disease and talked to them. And the theme of it was heterogeneity. Like every patient is a whole new story. And it was very impressive. And, you know, I recommend watching some of those videos that Michael J. Fox makes.
Okay then, why don't they ever do that surgery on both sides of the brain at once? Too risky? Bad outcomes?
So I have seen bilateral deep brain stimulation surgery take place. And so I'm not aware of, I'm not necessarily aware of it being riskier than unilaterally. One could imagine that it's a conservative approach to work on one side of the body and brain first and see what kind of benefit a patient gets before treating the other side. And I do want to give the same caveat that I am not a neurologist, but I have done research in a neurological setting and collaborated with neurologists.
It was the lesions that weren't done on both sides. The deep brain stimulation is commonly done on both sides, but the lesions were, there have been some bad outcomes with bilateral pallidotomy. And there had been, in the 1960s, there had been trials with pallidotomy made. And maybe because they weren't as good at placing lesions back then as we are now, they had had some very bad outcomes with patients who became psychotic from those lesions. And so it was considered, you know, I think the lesion treatment didn't get much of a chance to develop and to try various variants of it because it got superseded by deep brain stimulation pretty early on. And so maybe there were lesion treatments that would have worked that nobody knew would have worked.
So to clarify, Charles is talking about pallidotomy, and I was interpreting surgery to mean subthalamic nucleus deep brain stimulation surgery.
Okay. What kind of research or studies have been done using
No, I didn't select that.
that stuff on
That's good.
patients
Keep
with...
it. It's okay, we'll try it.
Do you know what that is?
I know what it is.
Oh, okay. That's why you're smarter than I am, yeah.
Yeah, I believe there's been, I think that there have been people who've tried that on their own, but I don't know of a proper clinical trial.
Yeah. What is it? What is the...
It's a hallucinogen.
Oh, okay. Sure. Yeah. Yeah, I'm out. Okay, once you have an animal model, why bother with the predictive model? Can you just try stimulation in various parts of the brain proactively?
The animal model that we like to use is the macaque monkey. And they're expensive, and they are enough like us that we really don't want to waste them or just try things for no reason. I mean, there's a lot of reservations about doing these kinds of experiments. You take a perfectly healthy monkey and give them a really serious neurological disorder that they wouldn't have had otherwise. And so they're kind of humanitarian concerns.
Yeah.
And so we're almost as careful with what we do in monkeys as we do with people. I've sometimes hear people complain that we are more careful with monkeys than we are with people. So it is true that an animal model could be used for more or less random experimentation, but we don't do that.
Yeah. I would say also there's a lot of parts of the brain.
It would be a random, it would be a lot of experiments.
Needle in the haystack, right?
The brain is a very large place.
Is there a test to detect early onset Parkinson's, perhaps analyzing neurological mechanisms, not considering cost constraints?
So I guess I can address part of this with highlighting some recent research results. So I'm aware of one study where patients have called in and essentially left a voicemail message, and then the fine patterns in their vocal patterns were analyzed, and this was compared between people who had Parkinson's disease and those who didn't. And it was informative. There was something that you can't detect by ear that I believe was different between those two groups. I believe there's also research in the direction of measuring gait more precisely, technologies like putting inserts with sensors into the shoes. There's a whole variety of technologies for taking kinematic measurements in an attempt to get earlier quantification of some differences in movement. Can you think of additional?
No, I don't know anything more than that.
And just to mention, these are not these are in the research stage. They're not proven diagnostics.
All right. So I just feel like this next question came from a UT neuroscience major. I just sense this. Does the death of dopamine neurons in the substantia nigra affect reward-based learning mechanisms in the brain?
Can I answer that? I love this question. So if you are a UT student who asked it, then thank you. So, the other thing that we know that dopamine does is, I kind of left you with the idea that what dopamine does in the striatum is mysterious. It was supposed to make one set of neurons fire more and another set fire less, and why would it do that, and how would it know when to do that, and all of that. I left all that part out. And we don't know everything about that. But one of the things that we do know is that, at least in our animal experimentations, it seems that the dopamine neurons are involved in the choices between movements on the basis of value. So what works? The dopamine neurons somehow get signals about what was successful movement and what was unsuccessful movement. And there's another set of dopamine neurons that don't control movement that are involved just in judging success and failure in a more general way, reward and punishment. But the ones in the substantia nigra seem to be engaged in the reward-based learning, right, of movement. And so one would think that maybe in Parkinson's disease there would be disturbances of reward-based learning of movement. And there are people looking for that and studying that, and there's a literature on it. It's a very interesting thing and surprising that on the face of it, Parkinson's patients don't seem to have any particular deficit in reward-based learning.
So if I can just jump in, because I'm channeling my mom and her Parkinson's. When she over-medicates with L-Dopa, she shows sort of the opposite. She does too much. And in fact, on the warning label for L-Dopa, among the side effects are excessive gambling. I was reading these to my mom years ago. Excessive gambling, shopping, and then it said hypersexuality. I skipped that one. But yeah, I mean, too much L-Dopa sort of makes the basal ganglia say, hey, let's do everything. Let's do too much. Okay, so, okay.
Yeah, I think that's a
So
important insight.
I've just seen my mom do that. So Charlie and Diane, we have this fun thing we do at Brainstorms. We have just a few minutes left, and so this is lightning round. And so the game is, can you answer these questions in 20 seconds or less, okay? We'll get to as many as we can. So here we go, lightning round. How can we apply AI, machine learning, in creating effective models for better diagnosis of Parkinson's?
So I don't know if this is AI and machine learning per se, but I am aware that, for better diagnostics. I was going to say for treatment, there are updated models for trying to create directed stimulation, so more advanced techniques for closed loop stimulation, and also trying to focus, get better control over the area of the brain where the stimulation is being targeted. And that involves computer simulations and data analyses along the lines of machine learning and AI.
What does this dopamine do anyway?
Dopamine controls changes in synaptic strength and synaptic plasticity. That's the thing that is most robust about the dopamine literature. It doesn't make cells fire more or less so much as it does control cells learning things about their inputs.
Beautiful. Could you discuss latest thoughts about alpha-synuclein in 20 seconds?
Okay, alpha-synuclein is a molecule that is overexpressed in dopamine neurons and in a bunch of other neurons that are affected in Parkinson's disease. Some people think that alpha-synuclein is the actual cause of Parkinson's disease. And the most important thing to know about alpha-synuclein right now is it actually the cause of Parkinson's disease? Because if so, that would be our first real handle on what causes cell death in Parkinson's disease.
Beautiful. Let's do two more. If Parkinson's disease and Alzheimer's disease both involve dopamine pathways, are there any treatments that target both simultaneously? I'm assuming AD
Some
is...
Alzheimer's disease patients have Parkinsonian symptoms, and those Parkinsonian symptoms often respond to normal Parkinson type treatments, even though the Alzheimer's disease symptoms mostly don't.
Beautiful.
I also know that deep brain stimulation is under investigation as a potential therapeutic for Alzheimer's, although I believe that the area of the brain that would be stimulated is different than for
You just
Parkinson's.
answered the next question. So one more. That was amazing.
But also obsessive compulsive disorder
Yeah.
and depression are other examples of disorders that are being treated with deep brain stimulation.
Last question. Is the DBS similar to transcranial magnetic stimulation used to treat depression?
Deep brain stimulation is much more precise, and it's delivered in a more targeted fashion, whereas transcranial magnetic stimulation, it's diffuse, it's stimulating larger areas of the brain. It is non-invasive, it's outside the head. I would say that both approaches are being studied in depression as well as in Parkinson's, but one's more targeted than the other.
Great. Well, we would do this all night, but Charlie and his wife have some visiting to do in Austin, so let's thank Diane and Charlie for a great time. So thank you again for coming. We'll be doing another one in November and December. You can go to utbrainstorms.com to see our schedule. You can, there's an email address there, which is mine. We love feedback. Tell us what you liked, what you didn't like. Suggest topics, all of that. We love to get your emails. I try to answer as many of them as I can, so thank you for that. And then I have to say that the kindest thing, if you like what you saw tonight, the kindest thing that you can do for us is tell other people and bring a friend when you come back next time, because that's how we've grown from 80 people in the audience to 400, 500, 600. So thank you for that. Be safe, and we'll see you next time.